Hello, everyone, and welcome to today's webinar, Next Generation Ultra-High Content Analysis of the Tumor Immune Microenvironment using mixed technology. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Stefan Eulitz, the Global Customer Liaison Manager at Miltenyi Biotec. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Stefan, you may now begin your presentation. Welcome.
Thank you for your participation in this webinar. Today I would like to show you the next generation of ultra-high content analysis using the mixed technology. To demonstrate the unique capabilities of the mixed technology, I would like to give you some nice insights into the composition of the tumor immune microenvironment. This structure is a perfect example to explain why it is really important to have the most detailed analysis possible due to its complexity and variability. But let's start with a short introduction to the topic. The solid tumor is embedded in the surrounding structures of its location. The complex system of different cell types, the extracellular matrix, molecules such as chemokines, cytokines, microRNAs, and other factors, as well as vessels, define the specific tumor microenvironment. Even by just looking at all the involved immune cells which are composing the specific tumor immune environment, it becomes clear that the characterization of all the cell types needs many proteins to be analyzed. One important proportion of the cell types involved are of course the immune cells, as they provide us with important information on, for example, prognosis and therapy of the individual cancer. The composition and spatial distribution of these cells define the tumor immune microenvironment. It can be classified based on the composition and the infiltrating cells, as well as the state of inflammatory response. If a tumor is populated with immune cells, but the tumor core is relatively void of cytotoxic lymphocytes, the type is classified as infiltrated excluded, which is shown in Figure A. We will have an example of this type later in the presentation. The second type is the infiltrated inflamed type, depicted in Figure B. This type is characterized by an abundance of PD-L1 expression on tumor and myeloid cells and highly activated cytotoxic lymphocytes expressing, among others, PD-1 and interferon gamma. The presence of a tertiary lymphoid structure, short TLS, adjacent to the tumor defines the third type, the infiltrated TLS type. A TLS is an aggregation of immune cells with a similar composition as lymph nodes, including B cells, dendritic cells, and regulatory T cells. We will also have an example of this type in the presentation. Given the complexity and different types of the tumor immune microenvironment and also the tumor microenvironment as a whole, the question arises, how can this structure optimally be analyzed? So what do we need? So first of all, we need to visualize a lot of different structures, cell types, and subpopulations. So this requires the possibility to stain a large number of markers. To illustrate that, so for example, you want to detect all different tumor cells and stromal cells, including immune cells, which obviously already requires many markers. Furthermore, in order to get a comprehensive understanding of the involved cells, it is wanted to analyze as many markers as possible to not miss an important characteristic of certain cells. Therefore, the number of markers should be even increased, and we have not yet addressed markers we need to visualize the tissue architecture. So it becomes very clear that a really large number of markers is needed. In addition to the cellular composition of the time, it is also important to understand the interaction of cells and the surroundings, which require information on the spatial distribution of the cells and tissue architecture. For this, it is necessary to have a technology which has not only cellular but subcellular resolution, as the intracellular localization of proteins, which means localization in the nucleus, in the cytoplasm, at the membrane, etc., will give you information on, for example, the activation status and so on. Light microscopy techniques are very well suited to fulfill this requirement, but standard microscopic approaches usually suffer from the limitation that the number of markers which can simultaneously be analyzed is too limited to really provide detailed phenotypes of all involved cell types and structures. The solution for this is the MIX technology and the MACSima instrument, and I will explain to you why. The term MIX means MACSima Imaging Cyclic Staining Technology. And this describes the approach of iterative cyclic staining processes as opposed to conventional single staining methods. This technology overcomes the conventional limitation in the number of markers which can be analyzed, and I will give you an explanation of the process on the next slide. This way of data acquisition is executed by the MACSima instrument, and only by the MACSima instrument, as there are several aspects which make the MIX technology and the MACSima instrument unique compared to other existing cyclic staining methods. The MIX technology uses conventional immunofluorescence stainings to visualize markers, and the MACSima is equipped with a fully automated epifluorescence microscope inside, which provides the resolution needed to understand the composition and structure of, for example, the tumor immune microenvironment. Let me shortly explain the process and what you will get as a result. So, first, the MACSima will start with a staining step. The resulting immunofluorescence signal is acquired, and then the signal will be erased. After that, the next cycle will start again, with a staining step, followed by the image acquisition and the erasure of the signal. It is important to note that only the markers will be stained in a cycle which are supposed to be imaged in the respective cycle. This has some advantages to prevent steric hindrance, but it is also much easier for generating image stacks with more than 100 or couple of hundred markers. Finally, you will end up with a large and unique image stack. But this is not the only advantage of the MACSima. The MACSima instrument is part of a complete imaging platform which enables you to analyze hundreds of markers on a single sample in a fully automated fashion, which means apart from loading the samples and the reagents, no manual steps are involved after starting an experiment. Furthermore, all necessary components to perform such experiments will be provided, for example, sample carriers, antibodies, and also analysis software. I would like to mention here that the MACSima platform is an open platform with regards to antibodies, which means that you are not dependent on the antibodies Miltenyi Biotec is providing, although we have a huge portfolio of high-quality recombinant antibodies. Indeed, we have the largest antibody portfolio compared to all other multiplexing technologies on the market. This complete platform solution therefore opens up such complex analyses to a very broad spectrum of users, as the technical hurdles are really low. It is also very flexible, as it can handle any kind of fixed samples such as tissues, adherent cells, or suspension cells. The preparation of the samples is almost not different to the current methods, and therefore does not require laborious and complicated steps, resulting in really an effortless experiment preparation. Another good message for you: the MACSima Imaging Platform is readily available now, which means if you are immediately interested, you can simply buy it. But let me convince you about the technology first. So let's look at some images. The topic for today is the characterization of the tumor immune microenvironment. And as mentioned before, this is a perfect application to demonstrate the need for the MIX technology and the MACSima Imaging Platform. There are two main aspects I would like to focus on today. First, I would like to show you a tertiary lymphoid structure. Afterwards, I will characterize the different immune cells on a specific type of a tumor immune microenvironment. So let's start with the TLS and a short introduction. The tertiary lymphoid structure is described here in further detail in this schematic representation to better understand the following images. So it shows the TLS and it is located within a tumor with a CD3 positive T cell zone containing dendritic cell lysosome associated membrane glycoprotein, short DC-LAMP, dendritic cells, fibroblastic reticular cells, and a CD20 positive B cell zone with a germinal center, plasma cells, antibodies forming immune complexes with tumor antigens and follicular dendritic cells. So the coordinated actions of CD8 positive cytotoxic effector T cells and B cells generated in the TLS enable in situ tumor destruction via direct tumor cell killing, for example, antibody-dependent cellular cytotoxicity mediated by macrophages and/or natural killer cells, and local complement activation. So central memory T and B cells generated in TLSs circulate and protect against metastasis, and in some cancers, TLSs located in inflamed regions adjacent to a tumor may also serve as a niche for tumor cell progenitors or tumor cells that drive relapse. So it is a really important structure to better understand a tumor. And today I would like to show you a TLS in an ovarian cancer sample which we have analyzed with in total 96 markers. So what can be seen in the data? Here you see the first ovarian cancer sample which I would like to show you today. You see the tumor tissue represented by the EpCAM signal in green and the associated leukocytes marked by CD45 in red. The nuclei are depicted in blue and vessels are shown by the CD31 signal. The massive accumulation of leukocytes adjacent to the tumor already indicates that this could be a TLS. So let's have a closer look at the real composition of the structure and whether we can prove this assumption. In this image, you see the distribution of B cells in this structure, and indeed, we have a B cell zone indicated by the CD20 signal, which is already mentioned as characteristic of a TLS. The next cell type we would need to see are the T cells. This is the staining of T cells with CD3 depicted in light blue, and we can see that many of the leukocytes are actually T cells. Now we need to see which type of T cells we have here. Here you see the two major subpopulations of T cells. The CD4 T cells, which are the main population here, are depicted in blue, and CD8 positive T cells are shown in red. We can see that both types of T cells are present in the TLS as expected, but we can see that the CD8 cells are also within the tumor tissue if we have a look at the very right tumor part. In this image, we can have a look at the proliferating cells, which is shown in white by the Ka67 signal. We can see that we have proliferating cells in the TLS, but also within the tumor. Another cell type which is missing but expected in a TLS are plasma cells. So plasma cells we have here detected with CD138 in white and CD38 in red. We can see that we have some few plasma cells in the TLS, but many others have also infiltrated the tumor area. By the way, this seems to be associated with a better prognosis in ovarian cancer. So now we have already B cells, plasma cells, different type of T cells, but there's another cell type missing which is essential for the TLS. These are the dendritic cells. In this image, you see the dendritic cells represented by the CD86 staining in orange. We can see the zone where we have DCs located. It has been stated that these cells should also express DC-LAMP or CD208. So the CD208 or DC-LAMP marker can be seen here in light blue. We can see that not all of the CD86 positive cells are also expressing DC-LAMP, which could also be expected as it is almost exclusively expressed on mature DCs. What you can also identify is that the DC-LAMP signal is not localized at the cellular membrane, but more in the cytoplasmic region. This is also as expected, since the protein is mainly localized within the lysosomes. This is a good example to prove that the previously mentioned requirement on and capability of the MACSima instrument providing subcellular resolution is true. So since DC-LAMP also plays a key role in the processing and presentation of antigens, this staining gives us the hint that the presentation of tumor antigens is also taking place in these regions. The staining of HLDR, here visualized in violet, is proving the existence of MHC class II receptors, which is also in line with the schematic representation of the TLS shown in the beginning. So one cell type which is not part of the TLS but of the tumor microenvironment are myeloid cells. These cells are shown here in yellow by the staining of CD14. The staining of CD163 in white shows us here that several of the CD14 positive cells are tumor-associated macrophages. We have now seen the major immune cell types important to define the TLS, but I would also like to share with you an example which demonstrates that the findings do not have to be restricted to the identification of certain cell types. So here you can see MICA79 in red, and this is a marker for high endothelial venules, here abbreviated as HEV. I would also like to show you again the CD31 staining of vessels from the beginning, here in blue instead of white. And as you can see, not all vessels express MICA79 and are HEVs. To conclude this part, I share with you this overlay of 7 markers illustrating the architecture of the TLS, which we could identify and prove due to the numerous markers stained using the mixed technology and the MACSima. So let's move to the next example I would like to show you today, which includes also many findings from the last years and proves the power of the technology, since you can see all these great work done in the past just in one experiment. You will now see a second ovarian cancer sample where we have characterized the tumor immune microenvironment. As said, this is another sample of ovarian cancer, and as already done for the first sample, the tumor is here depicted in green, leukocytes are depicted in red, vessels in white, and nuclei in blue. Additionally, also fibroblasts are depicted in blue. It is immediately striking that the CD45 positive cells are mainly localized in the invasive margin of the tumor and not in the tumor core. This could show us that we have a tumor type of the infiltrated excluded type, but let's look closer at the phenotypes of the cells. In this image, you can see the plasma cells in white represented by the CD38 signal. And these cells are also located in the invasive margin and absent from the tumor, as could already expected from the initial stainings. So here in magenta, we see the T cells, and these cells are mainly localized in the invasive margin and almost completely absent from the tumor core. This underlines the assumption that this tumor is of the infiltrated excluded type. But let's see the different populations of the T cells. So when we take a closer look at the T cell populations and their localization, it is even more obvious that cytotoxic T cells are not in the tumor, but stuck in the tumor margin indicated by the CD8 staining in red. The few CD3 positive signals within the tumor core are only CD4 positive. So the myeloid cells are also mainly located in the tumor margin, but some CD14 signal in yellow can also be detected in the tumor core, as you can see here in the picture. Also, granulocytes can be detected in the tumor. here represented in light blue by the CD15 signal. So here you can see all the cell types in one image, which have already been described, and this supports the designation of the infiltrated excluded type. So in this picture, the macrophages are missing, but since they play an important role, we will take a closer look at them. So tumor-associated macrophages are a critical component of the tumor microenvironment, since they can affect growth, angiogenesis, immune regulation, metastasis, and chemoresistance. So macrophages are shown here in red by the CD68 signal together with the CD14 signal in cyan. We can identify two different localizations in the tumor margin and within the core. It is known that most of the macrophages gather in the leading edge and avascular areas. Others can align along the abluminal side of the vessels. So let's see whether we can also see something like this. So here you see macrophages in red and vessels in white. Indeed, some of the macrophages seem to be located along the vessel in the tumor core and in the tumor margin. So in an attempt to further characterize the phenotype, we see here CD163 in light blue, which is a molecule on macrophages involved in clearance and endocytosis of hemoglobin-haptoglobin complexes. Furthermore, this molecule is mainly present on M2 macrophages. We can see here that there are already different macrophage phenotypes present, as not all are positive for CD163. So in this picture, you can see CD206, which is depicted in yellow. This is another marker present on M2 macrophages, which could also serve as a prognostic marker. As not all macrophages are CD206 positive, it is quite obvious that we have here different subpopulations of macrophages. I will not go into more details regarding the macrophages and their phenotypes, as this would go beyond the scope of this webinar, since this topic would justify an own one. But I hope that I could present to you two nice examples which give you a flavor of the technological capabilities of the mixed technology and the MACSima instrument, and of course also the potential for your research. Although we have seen more markers on one sample than possible with conventional technologies, we are not really close to the full potential of the mixed technology. Therefore, I would like to give you a quick insight into what else is possible apart from just nice images. So I will now illustrate the data workflow of how it would be if you were characterizing the tumor immune microenvironment with a mixed technology in your lab. So everything would start with the data acquisition done by the MACSima instrument. And this is then, of course, done in the cyclic staining process. And then you would obtain an image stack of the markers you have used. Especially in the beginning of a project, you would most probably start with 100 or even more markers in order to start the project as unbiased as possible, as any non-stained marker would be self-chosen limitation. Since the MACSima can image hundreds of markers, there is no technological limit existing. So after the quality control of the images, you can then start with the segmentation of the images. This can be done with our analysis software, which is QI Tissue, or with any other software you are using, as the images are available in an OMI-TIFF format. After that, you can extract the data, and this allows you to go much deeper into the analysis. And for this, I would like to give you now a first impression. So here you see a third sample of an ovarian cancer. And in this case, the sample has been analyzed with 98 markers. I hope that the transmission of the video is good so that you can see all the marker images. It is obvious that there are many different staining patterns and regions of different composition, and this can be immediately identified. Having the potential of also including markers which are supposed to be negative on the sample is also a huge asset of the mixed technology, as you are not really limited in the number of markers. This is also a very good opportunity to verify existing assumptions in an easy and comprehensive way. Nevertheless, such data sets are complex and need some initial orientation. In order to help you to get an overview, I will briefly show you the composition of the sample with the most important markers, as you have already seen for the other sample examples. So as you can see here, the tumor area is again indicated in red using the EpCAM signal, and the leukocytes are shown in red. The vessels can be seen in white and fibroblasts in violet and the nuclei in blue. We see very well that there are a lot of leukocytes adjacent to the tumor, and it is of course worth spending another look and a deeper look on the sample. So as you can see here, the tumor microenvironment is composed, of course, of different immune cell types as expected. So in this case, these are predominantly myeloid cells indicated by the CD14 signal here in blue, and also the T cells represented by the CD3 signal in red. But also we see some plasma cells, which are indicated by CD38 positive and CD138 positive cells. So here in this example, we see some other cell types. So the tumor microenvironment is not only consisting of T cells and myeloid cells, but we can also see some natural killer cells shown by the CD56 signal in red, and also some regulatory CD4 positive regulatory T cells, which are here shown with the signal of CD25 in blue. Also some very few dendritic cells represented by the CD1C signal can be positive. So now you have gained at least first overview on the cell types we can find here in the sample. But when we look at the markers we have shown, these were only 12 markers. But as you could see in the beginning, we have stained many, many more markers. So in order to be able to analyze this, I will show you the next necessary steps here. So the next necessary step is, of course, the image segmentation. And here you can see the segmentation mask of this image. And in this, we could identify in total 4,425 cells. So the segmentation can be carried out by different ways. So you can use our QI tissue software, but also any other algorithm can be applied, as these images are available as OMITIF files and therefore accessible for any kind of analysis software as mentioned before. So segmentation is the prerequisite for data extraction, and this is then opening up the opportunity to analyze the dataset as a whole. So when you have extracted the data, there are many different ways to analyze it. You can use our QI tissue software, but since the data table can also be exported, any type of analysis is possible. So here the result of one option is depicted. So in this case, the cells were first hierarchically clustered, which is not shown here, and then the main groups of cells were determined based on the expression patterns and given a color. After that, the dataset was visualized in a t-SNE plot. So what we can see here is that we find all the major groups of cells, which were also shown in the overlay images in the beginning. But there are also some unclassified cells which might need a closer look and could bear some striking findings. But this is, of course, just the surface of the whole information in the dataset, as we have clustered based on 98 markers. So furthermore, the X and Y coordinates are available, and therefore the localization of each cell is also known. And this can also be depicted in the image. This also enables the analysis of interactions, distances, etc., which could also provide useful insights into the biology. This should just be an outlook what can be done, and we will show more like this in the near future. Therefore, have a look at our coming webinars in order not to miss any information about it. We are now at the end of the presentation, but I hope I could show you the capabilities and the potential of the mixed technology, and that it is absolutely amazing to have so many findings in just one experiment. If you are interested in more information about the mixed technology and the MACSima Imaging Platform, do not hesitate to contact us via our websites or sales reps in the field. And again, the good news is that the MACSima Imaging Platform is now available for buying. So with this, I would like to thank you for your attention, and I hope that you have enjoyed the webinar. Furthermore, I would also like to thank our teams in research and development for their hard work, and I'm now happy to answer all your questions.
Yeah, Effie, get your butt inside.
We'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. All right, let's go ahead and get started. So your first question, what should be the thickness of section?
Yeah, so basically, since this is an epifluorescence microscope, the thinner the better. So depending on the type of fixation, you can start with maybe 3 microns going up to 10. So on average, we are using 5 micron slides.
Great, thanks so much. Now, is it possible to analyze full tissue section or only region of interest?
Yes, it's definitely possible to analyze full slides. That's not a problem. Of course, these are huge data sets, so you have to handle those data, but technically this is not a problem. Of course, such runs are taking a bit longer, but there's no limitation in terms of regions of interest. So whole slides can be analyzed.
Thank you. Can we use any other desirable marker?
Yes. As I mentioned in the presentation, the platform is an open platform regarding the antibodies. So there are just some prerequisites to consider. You need primary conjugates of an antibody. So that means we need, in this case, primary antibodies coupled to a fluochrome. And these fluochromes would have to be bleachable, because this is one of our erase mechanisms we are using, and therefore this is something you have to obtain. But if you have this is not a problem. You can simply use any kind of antibody you have and generate a conjugate out of it, and that's it.
Great, thanks so much. Now, this next question I saw, a few times. How long does it take on average a staining and imaging cycle?
Yeah, so here multiple factors are, of course, contributing to the cycle time. Of course, the size of the region of interest is important, but I can say that so the shortest cycle will be roughly 30 minutes, maybe some minutes more, but this can go up to several hours if you are, for example, analyzing a whole mouse brain or something like this. So this is the range you can expect.
Great, thank you. Okay, let's see. Our next question is, the presentation did not include antigen staining that requires antigen retrieval. Is MIX compatible with antigen retrieval?
Yes, so you can use basically all kind of fixed tissues, so FFPE tissue, which involves such an antigen retrieval protocol. And here, the most important thing is that all the antibodies you want to use are compatible with the retrieval method you are using. So you are not obliged to use just one. If you can make sure and test whether the antibodies you want to use are compatible with this method, you can use it. So our antibody portfolio has been tested on the retrieval method using tech buffer pH9 and heat antigen retrieval. So for this protocol, we can tell you which antibodies from us are working, but that doesn't mean that this is the only protocol you can use. So yes, the MIX technology and the MACSima instrument are compatible with fixation methods needing antigen retrieval.
Thank you. Now this next question says, what is the manual logistics in loading or preparing 100 antibodies for use on the MIX platform to be utilized all 100? And the second part says, can it store the number of stock antibodies for later staining workflows?
So basically for this, you would have to prepare a 96 deep well plate, which we are using for as a reagent carrier. So you will simply pipette these antibodies into those wells, and that's basically it. But if you want to skip this step, you can also purchase from us ready-to-use plates, the rear screen plates. And then you can even skip this process or this process step. And this is then really almost without any needed preparation. So you have full flexibility generating your own plates or using our ready-to-use plates. But it's not really complicated. It's just pipetting antibodies into a 96 well plate.
Thank you. We're receiving such great questions from the audience. Now, this next one is a two-part question. How are the colors distinguished and how reliable are the acquisitions?
So we are using filters, of course, for excitation and then also for emission. So we are separating the signals this way. And the colors you have seen are, of course, artificial colors. We are just detecting grayscale. So we have optimized our filter settings to basically be able to image the entire spectrum, but for now we are starting with 3 fluorochromes, FITS, PE and APC.
Thank you. Now a reminder to our audience, any questions that we do not get to today will be addressed by Stefan via email following the webcast. And let's go ahead and move on to our next question. Is data analysis going to be challenging here?
The simple answer is yes. On the other hand, there are nice tools to do so. So our analysis software, QI Tissue, is really making your life much easier there because it is optimized for visualizing such kind of data sets and also analyzing. So you will have the option to segment the cells. The picture you have seen is also generated with this platform and this software. And then after segmentation, of course, it also offers some analysis algorithms like t-SNE plots and k-means clusterings. And this will, of course, also evolve over time. So yes, analysis is challenging, but the tools we are providing are also making your life much easier so that you can handle these data sets quite well.
This is a great question. What is the distinction between your platform and other multiplex platforms, such as multiomics on the market?
Yeah, so the major differentiator here is, of course, the number of markers we can stain. Yeah, so as I mentioned in the presentation, we can go up to hundreds of markers, which is mainly due to the fact that we have a very mild process of erasing these signals so that we can obtain several hundred marker images. This is not a problem, and this is the main differentiator, but there are also some other details which are different. So the way we erase the signal, for example, and also the antibodies we are using, this is also a bit different, and I think the fact that we are offering a full package, so you can get everything from us. Of course, you have the option to add something to this, but you can get everything from us, so you don't have to worry that you have to solve a problem or something like this. It's really, I wouldn't say plug and play, but it is really an easy solution for everybody.
Stefan, can you tell us how you erase the stain and signal?
Yes, so basically we are using two different methods. One method, which I already mentioned with the question of the antibodies from a third party, we are using bleaching. This is a very nice tool because we have optimized this bleaching. We have a dedicated bleaching unit in our system. So this offers us also the option to open the platform also for other antibodies. But there's a second one, which is our new erase mechanism, and which is even more gentle to the sample than our bleaching. And this is based on Miltenyi Biotec's Release technology. So we are calling this Dialys because this is a controlled release of the fluorochrome. We are using specialized antibodies for this, and this is even, as I already said, more gentle, and especially for large samples also faster. And this is the erase mechanism of the future.
Thanks so much. Next question, is this method restricted to particular tissue types, or can it be used for any tissue type?
So you can use it for basically any tissue type. So we have tested basically any kind of solid tissue, tumors, and so on and so forth. The most important thing is it is only restricted to fixed specimen. Yeah, so you have to fix it either with, for example, PFA or acetone, or as I already mentioned, FPE. Therefore you can basically analyze everything, but not only tissue. You can also analyze adherent cells or also suspension cells. For this we are also offering a solution so that you can have a single cell analysis on suspension cells.
Thank you. Now, how do you compensate for autofluorescence?
Yeah, so here we have a nice advantage. So after or prior to a new cycle, we are acquiring images which we can then use as a background and are subtracted from the marker image. And with this, we can approach the problem of autofluorescence. So you can also image tissues which are usually suffering from a lot of autofluorescence, for example, liver tissue, because this subtraction offers you still the dynamic range, which is basically the difference between the potential of the camera and the signal intensity of the autofluorescence. And with this, you get very nice results also with tissues having high autofluorescence signals.
Wonderful, thank you. Now, is it possible to rule out the possibility of cross-reactivity of various antibodies?
Yeah, so we have here some process-related advantages. So if you were staining several tens or maybe 20, 40 antibodies simultaneously, I would say, yeah, there is quite some risk that there is some interaction or competition between those antibodies, since we are just staining every antibody in the cycle which we want to acquire only in this cycle and not directly in the beginning. We do not have faced such an issue. The only thing I can say is if you are imaging markers with a competing epitope, so an overlapping epitope in, let's say, the next cycle, there you can see that there is a blockage of the epitope because the antibody is still on the sample. But if you stain this antibody with the overlapping epitope in a later cycle, this is possible because the antibody will detach because of the washing steps and so on and so forth. And therefore, for us and our technology, this is not a problem.
Great, thank you. Can we identify intracellular markers?
Yes. So an important point here is to mention, I mean, if we're analyzing tissue slices, of course, and they are fixed, so the fixation method is also generating holes into the membrane and so on and so forth. So yes, you can see them, but you have to be careful if you would like to visualize soluble factors. Make sure that you are using a fixation method which is cross-linking these molecules. So you should not use acetone fixation, for example, for those factors, but rather PSA, for example, or FFPE, and then you can definitely image those markers.
Thank you. All right, your next question is, can we process paraffin section or cryosection compulsory to utilize this imaging system?
So, the processing of the sample is, of course, done outside of the MACSima. So the MACSima is performing every step fully automated, but the sample has to be processed outside. So yes, you can process these samples, frozen sections, paraffin embedded sections. You just perform the processing step outside of the MACSima and then put these samples into the MACSima, and then the MACSima Imaging Platform is doing everything for you, fully automated.
Thank you. Now, can we quantify TILs?
Sorry, can you repeat that?
Sure. Your question says, can we quantify TILs?
Yes, you can quantify tumor-infiltrating leukocytes because you can, first of all, easily identify them because you can stain a lot of markers. So therefore, you can really be sure that these are tumor-infiltrating leukocytes. And the resolution is, of course, standard immunofluorescence resolution. And therefore, yes, you can.
Wonderful. Thank you so much. We are receiving so many great questions from the audience. Now let's go ahead and jump over to this one. And it says, can one converted imaging data to a statistical data be used to compare, for instance, a carcinoma to an acquired resistance form of the same carcinoma?
So yes, you can extract, of course, after segmentation of the sample, which is a prerequisite here to have, let's say, all cells identified. You can extract the data here, and then all statistical options are open to you. Yeah. So therefore, yes, you can do this and use every kind of statistical method you need to answer your question.
Now, how many markers can you view per round?
So at the moment, the number of markers is 3 per cycle, plus the DAPI signal. But I have to mention that the optical bench, so the microscope of the MACSima, is already equipped to cover the entire spectrum. That means there will be new fluorochromes coming out, especially with a new erase mechanism based on our release technology. And there you will get new fluorochromes which do not need to be bleached. And here we will cover in the future the entire spectrum, so this number will increase.
Now, how do you fix the tissues for staining protocol? Do you recommend any particular reagent?
So basically, we have tested our entire antibody portfolio on 3 different fixation methods: the FFPE with the epitope retrieval protocol based on pH 9 tech buffer and heat, PFA, and also acetone fixation. So if I have to recommend you a fixation method, that is of course very much dependent on the application. As I already said, soluble factors, if you want to analyze those, you have to use a cross-linking fixation method. But it is also dependent, let's say, on the antibodies and the specificities you need and the availability of antibodies. If I have all the antibodies available which I need for a certain fixation method, but for the others not, maybe then you would go rather with this fixation method where you have the antibodies, if you still can visualize what you need to know.
Thanks so much, Stefan. We have time for a few more questions. So next question, how much antibody is used per round per marker?
That depends, of course, on the size of your sample. So we are offering different sample carriers for standard microscope slides. So we are offering Maxwell disposables with 4 chambers, 2 chambers, and 1 chamber, depending on the size of your sample. You can expect costs between €3 up to €20 per antibody.
Great, thank you. And what is the processing time per staining?
So we are using here an incubation time of 10 minutes and then several washing steps, and this results in the end, let's say, in a minimum cycle time of 30 minutes. But then, of course, the size of the region of interest is then determining how long a cycle would be.
Thank you. Now, is it possible to utilize cocktails of antibodies in a single staining and imaging cycle?
Yes, this is actually what we are doing, because we are staining up to 3 markers per cycle. And in the future, as I said, this will be increasing. So therefore, yes, you can do that.
Thank you. Now, is it possible to take DIC or bright field images in parallel in each case?
Unfortunately, this is not possible. So the MACSima does not have, let's say, a bright field image or cannot generate bright field images. What is, of course, always used and acquired is the DAPI signal, so the nuclear signal. So therefore, this is something you can always use also as a control, whether, for example, the image acquisition has worked properly.
Thank you. And we have time for just a couple more questions. What is the maximum number of markers you can use with this technology?
Yeah, so basically I cannot tell you because we haven't reached the limit yet. So as I said, a couple of 100 markers is possible. This is what we have done, but since at that time when we started this, we didn't have more than 400 markers. I cannot tell you whether you can stay in a couple of hundred more, because we simply didn't have more. So of course, I have to say here that this is also dependent on the tissue you are using. So not every tissue will survive 400 antibodies or 200, but what I can say, all the tissues we have checked so far are definitely able to survive more than 100 markers on this specimen. Therefore, yeah, I'm curious to find out what is exactly the limit. Yeah, stay tuned also in the future. We will let you know where we think is the limit.
Thank you so much. And let's go ahead and wrap up with this final question. How do you make sure that the images from the different cycles are properly overlaid?
So as I already said, we are acquiring the DAPI signal every cycle, and this is what we are using to properly overlay the images, and therefore you can be sure that you will end up with a nicely registered image stack after the experiment. So this is basically our technological advantage to have one marker in every cycle.
Thank you so much, Stefan. Do you have any final comments for our audience before we go today?
Yeah, so I hope I could provide you further insights into the technology with my answers today. And yeah, let me just tell you that today we have just touched the surface of what is possible with the mixed technology and the MACSima Imaging Platform. I mean, with this technology, we are now entering into a phase where we will much better understand nature's complexity. And of course, in addition, we will be able to answer questions which we did not know that they existed. Therefore, there's much more to come. And again, watch out for our next webinars and our webpage, and contact us if you want to know more about this platform and the possibilities related to this technology. And yeah, I simply thank for your participation, and I will enjoy seeing you next time.
Thank you again, Stefan, for your time today and for your research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. As a reminder, any questions we did not get to today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.